Thank you very much to Dr. Mesa and the team for the invitation to come here. I was last here in 2019, and I remember leaving the conference with the biggest buzz, so happy to have met people, and looking forward to coming back. And we missed one meeting due to the pandemic, but I'm really happy to be here, so thank you for the opportunity. Right, so I have the unenviable task of explaining to you, well, first of all, what does my title even mean? Molecular biology. So whilst many of the talks here are going to be by absolute international experts in the field about what to do about MPNs, my role here is to tell you why do they happen, how do they happen, what do we understand about everything to do with MPNs before the patient presents to clinic. Okay, and molecular biology means at the level of molecules and proteins, what is actually driving the disease. So I'm going to start really basic, and then I'm going to go really advanced, and hopefully I won't lose too many people along the way. Right, this is blood. When we take blood out of our veins, this is what it looks like. And when we look under the microscope, and one of the reasons why I loved hematology and got into hematology is you can see the patient, you can actually then go to the lab and see the blood yourself. And so these pink things are our red cells. They carry our oxygen around the body. These funny looking ones with purple blobs in them, they're all our different types of white cells that fight infection. And then the teeny tiny things that you can barely see, which look like dirt on the slide, are platelets. And platelets help our blood clot. Now, when you look in the bone marrow, which is where our blood is made, it's like the factory of blood. All the mature, ready-made blood cells are out in our blood, but all the precursors, the things that aren't quite blood cells, are in our bone marrow. So we have the parents of the white cells, or the leukocytes, we have the parents of the red cells, and we have these ginormous cells, which are the parents of these teeny tiny little platelets. And they all exist in the bone marrow. And these little white blobs here, these are fat areas within the bone marrow. This is what it looks like. So it's a super rich environment, and as hematologists, we're used to looking at blood and bone marrow to try and understand, does it look normal, does it not? And what's really key is that when we are born, we start with 1 cell, 2, 4, 8, we become an embryo, a body, and there are key cells that are there with us for the rest of our life, and they're called stem cells. And stem cells make other cells, and those other cells do their job and then die off. But the stem cells stay there and are with you for the whole of your life. So what happens in these cells isn't really hugely important. If they develop a problem, they'll be gone in about a week. But what happens in these cells are really important, because if something happens to these cells, they're there with you for life. And the stem cells live in our bone marrow. And what happens in the MPNs is that the stem cells are overactive. And so rather than making the normal number of white cells and red cells and platelets, we're making too many red cells and too many platelets, depending on the flavor of the MPN that different people have. Now, are these conditions unusual? They've actually been around a long time. They've been described over 100 years ago. They're really old conditions that have been with humanity for a long time. But they've actually, in the sense of, the Darwinian tree of life, they've been around for a long time in other species. So dogs have them as well, which tells us that the causes of these conditions arise in proteins that really have existed within evolution of species for a long time. They're not in proteins that are unique to humans. They're in really bread and butter style proteins that are responsible for making blood that exist in all species. So these are the questions I'm going to address today, I hope. What caused it? These are questions that patients ask us. What caused my disease? Why is my disease different to the other person's disease? Why is mine benign, another person's more advanced? Why do I have symptoms but another person doesn't? What causes these differences? How long have I had it for? This is a question I get asked quite a lot, because people are trying to make sense of why now? Did I have it 6 months ago when I wasn't feeling quite so well? Did I have it 5 years ago? What happened in my life that meant that I then started to feel unwell and got this diagnosis? So how long have I had it for, and how fast did it grow, and is it still growing? These are incredibly important questions for our patients. And we're going to start one by one and go through them. And I'm just going to give you a flavor for each of these questions. So what caused it? Right. So within each of our cells, and we have about 30 trillion cells in our body, Within each of our cells, we carry chromosomes. Chromosomes are inside our cells, and they carry the code for making anything in our body. Okay, so these are our chromosomes, and this is actually a really lovely picture. This is the first time in history that we were able to count the number of chromosomes that we have in different species, including chimps, dogs, cats, goldfish. They all have different numbers of chromosomes. And chromosomes are really these lumpy structures that when you unravel them and open them out, you then have the DNA here that comes out. And chromosomes are a way of getting miles and miles and miles of DNA into the tiniest little package so that they can fit inside a cell. And so what we're going to talk about today is DNA, the code for life. And it's made up of an A, a T, a C, and a G. And the order of those tells you what proteins to make, how to make them, and the recipe. So within that DNA, that code, there are elements of that DNA, about maybe 2% of that DNA, that codes for making proteins and molecules that we need to be who we are. And one of those is called Jack2. And Jack2 is a protein that you wouldn't normally know of because it sits inside the cell. It's not floating around your blood doing things, talking to different organs. It sits inside your cell. And all it does is take a signal from outside the cell and it tells the cell what to do. And what happens in the MPNs is there is a change in that code for Jack2 that stops it from switching itself off. So whereas Jack2 switches on when there's a signal that says stem cell, make a red cell, your body needs some red cells, that's Jack2 switches on, takes that signal, you make red cells, then it stops. With the mutation in Jack2, the off signal doesn't work anymore. So it's constantly telling the cells, the stem cells, make more red cells, make more platelets. And it doesn't need to. But there's a change in that DNA code that happens by chance that means it's killed the off switch. So this is the off switch here, and it normally switches this off, which is the on switch, and this mutation, V617F, that many of you may recognize, and it doesn't matter if you don't, it stops the off switch. So what happens is EPO, erythropoietin, we hear about erythropoietin, Tour de France cyclists love erythropoietin. And erythropoietin normally goes to a cell and tells the cell, make red cells. And Jack2 shuttles and does that messaging. If that's on, then the cell thinks it needs to make more red cells, even if the body is switching off erythropoietin and saying stop. And so you get more red cells. So really, this was discovered in 2005. I remember I was a junior doctor back then trying to work out what specialty I was going to go in, and I arrived in hematology, and about 3 months before, everyone was talking about Jack2, and I thought it was J-A-C-K, and I was like, who's Jack? And then I realized that there was this, mutation. I didn't even know what a mutation was then, and it was quite exciting, actually, because up until that point, to understand if people were making more red cells because of some other reason, a lung problem or a kidney problem, you had to do so many tests, whereas now you could just do a test for the Jack2. And within 5 years, because of the most amazing clinical research community that we have and collaboration with drug-making companies, we had JAK inhibitors in clinic and the first reports in the journals, and these have now changed our practice. But if I go back one slide, in about half of patients, we still didn't know what was causing it. And I remember starting my PhD in the MPNs, and my boss then, Tony Green, who used to come and speak here a lot, was like, well, what's one of your research questions? And, you need a research question. And I said, well, I want to know what's happening in the other 50% of cases. He's like, yeah, well, everyone's trying to find that. And but it was a burning question in the field. And so we started doing sequencing of DNA, technologies that didn't exist really at the time of the discovery of Jack2, but they were coming into play where you could sequence every bit of DNA. And so we started doing that. And then, during that time, other genetic mutations, so changes in genes, were being discovered that when you didn't have a Jack2 mutation kind of did the same thing. It either switched something on that made your body make more blood cells, or it stopped an off button. So these mutations switch something on, make you make more platelets, and mutations in here stop the off button, and so you end up making more blood cells. But we still didn't know what was happening in 50% of cases. And then after sequencing the DNA, we discovered that the vast majority of patients that didn't have any of those other mutations, these patients in red here, had mutations in CALR. And we were just discussing this morning that, in July of this year, it'll be 10 years since that discovery, and it's pretty amazing. So this was a new test for in clinic and in patients, and then I sort of realized at the time that research does make a difference, because here we have now this new discovery. Within a month, we were testing it in all our patients in Addenbrookes. I still have the patients in whom we first detected it in our clinic today. And they came in at a weekend and gave us their blood samples because we wanted to make sure we weren't making a mistake. We have such an amazing community in MPNs. And the reason we can detect this mutation so easily, it changes the size of the protein, or the DNA sequence really, and so you can detect it. And what's even more exciting is the fact that 2 months ago at the American Society of Hematology, one of the drug companies, Incyte, presented data on the first ever CALR antibody, which is really the magic bullet. And first in human trials, which is where you start using the drug to see if it's safe in humans, commenced this year. So it's just amazing how much progress is being made with time and energy. So really, mutations in Jack2, CALR, and other genes such as MPL drive MPNs. They drive our body to make more blood cells. And most patients, but not all, and there may be some people here who don't have those mutations, most patients do have mutations in one of these 3 genes. But to bear in mind, a lot of patients and a lot of people who don't have MPNs have mutations in many genes. It's part of life. We develop mutations in many other genes, and some of you here, but maybe not many, will hear of mutations in DNMT3A, TET2. These are common as we get older, and as all of us in our room get older, we'll be able to detect these things in our body, regardless of whether people have MPNs or not. It's part of normal aging. So that's what causes it. So what causes the difference between individuals? So I've already talked about the different mutations. Depending on what mutation is present, you get a slightly different flavor. But even within the same mutation, it can cause different things. So we know that a higher level of Jack2, so if you have more cells that have the mutation in Jack2, you're more likely to go towards a polycythemia vera type phenotype. If you have double copies of the mutation, so if you develop one mutation, our body has this tendency to duplicate it. It's quite normal. And if we do that, we go down the route of making more red cells. Because if you have 2 mutant copies, the signal is about red cells. If you have 1 mutant copy, usually the signal is make platelets. We know that specific mutations in Jack2 will drive polycythemia vera versus others. And we also know that the order in which you get the mutations, if you get a Jack2 followed by something else, versus if you get something else followed by Jack2, it affects what disease you get. So in general, a lot of the differences between diseases is to do with the composition of the different genetic changes that are within each sort of blood cell. But it's more common, there are other things as well. It interacts with so many factors. It can interact with your age. As we get older, there is a predisposition towards making more red cells. Men have higher hemoglobins. They're going to have more of a risk of developing polycythemia vera. Women will often have essential thrombocythemia, but after the menopause, where you're not naturally losing blood, you're more likely to develop polycythemia vera. And then these funny numbers at the bottom here, this is our very easy way of referring to DNA changes that we're born with. I don't know why we do this. I mean, it's really not intuitive, is it? And so these refer to not mutations, but things we're born with, which is why I look different to the next person, because of all the DNA changes that I have that someone else doesn't have, that I got from my parents. And depending on what I have, it determines whether I'm more likely to make red cells or platelets. And if I'm more likely to make red cells, normally, if I get one of these mutations, I'm more likely to have polycythemia vera, and likewise. So it's a combination of mutations that we acquire with life. It's a combination of our gender, our age, and it's also a combination of the DNA that we're born with that influences the kind of disease that we have. In myelofibrosis, it's exactly the same, although additional mutations are more common. So what we've done in the lab in the last couple of years is we've developed this personalized prognostic tool, because people like to know what to expect in the future. And so we're developing a new website here. It's going to be called Predict Blood. It's going to be launched this year, where we took patients in the past who who we had profiling of everything about them, their age, their gender, their mutations, their labs, their blood lab tests. And we tried to understand, well, what happened to them based on what their criteria were at the start. And based on that, we try and predict what's going to happen to someone who we haven't yet seen the future, based on what's happened to people in the past. And we can use that to predict, and the different colors tell people the likelihood of different things happening in the future, just being well with their disease and it not bothering them, maybe progressing, maybe developing acute leukemia. And hopefully with time, this might be a tool that can help physicians understand who to watch out for and who not. Although we don't quite know, yet how best to use these tools, how best to use them to guide therapies, which is, the next stage. So the last two things, how long have I had it for and how fast did it grow? I think I'm doing okay for time. I've got another 20 minutes. So how long have I had it for? I think this is quite an important question for patients. And before I go into how we managed to do this, I want to take a little interlude. The challenge of making blood. So the bone marrow makes 200 billion red cells a day. And it makes 10 billion white cells a day, every single day of our life. And every time one cell is made, the parent cell has to divide into two cells. And every time you divide into two cells, you have to make a copy of your DNA, the entire DNA, and you have to pass it equally to both cells. And your DNA is 3 billion pieces of DNA, nucleotides, those A's, T's, C's, G's. So we have 3 billion nucleotides of DNA for one cell division, and we have to make, 210 billion cells a day. That's a lot of DNA copying. And mistakes happen. So mistakes happen when we're making DNA and dividing, but mistakes also happen just with time. When a cell is sitting around, its DNA is constantly being attacked by chemicals within the cell, by, UV light, by smoking, but in blood, it's not really those factors. It's mostly just things that are there normally. It's not like lung cells where smoking has a big impact, or skin cells where sunlight has a big impact. The blood is very well protected. And in fact, we think that when we were sea dwellers and moved to the land, blood went into our bone marrows to protect it from insults that might mutate the DNA. And fish do not make blood from their bone marrow. Fish make blood. I'm not going to digress too much into other species because I find other species fascinating, but fish make blood from the head of their kidneys. And they think that as we moved onto land, we had to protect the DNA in our blood because blood is the most proliferative organ in our body. So if we think of all the cells that we make every day in our body, 98% of them are blood cells, and maybe 2% are skin and gut cells. So it's a really important organ to protect. So if we know now that we acquire about 17 mutations in our 3 billion bases within every blood stem cell per year, and we have 100,000 stem cells. We know that too now. So all of us here have about 150,000 stem cells. I said to you earlier, 2% of our DNA is important and codes for proteins. So if we do the maths, 100,000 times, let's say 20 mutations a year to make the maths easier, that's 2 million mutations per year that we're acquiring. 2% of that is 20,000 mutations per year in important bits of our DNA. That's one mutation every 15 minutes. So I hope my talk is going to be worth the 2 mutations that we all will have acquired by the end of the talk. So this is normal, this is life, this is happening in all species and all of our tissues, and actually blood is very well protected. This is a very low number compared to other organs. And so DNA mutation numbers give away our age. The most accurate way in which we can tell how old someone is to take a single cell and measure the number of mutations that they have in any of us. In fact, we had one individual who had the mutation number of a 32-year-old, and they were meant to be 70, and we were like, wow, there's something interesting here. And we realized that the paperwork was inaccurate, and they were 31 and a half. So this graph on the left tells us age and the number of DNA mutations, and it's a clock. But, we don't worry about this. This is life. And we use it to our advantage because it allows us to time events. So if we know how many mutations we have, and if we know the order in which they occurred, we can then see when mutation 3 occurred or when mutation 5 occurred. And so we did this in patients with MPN. I'm sorry the slide's gone a bit funny, but we took 10 individuals from our clinic in Cambridge. All the different colors underneath them represent different diseases, different durations of disease. They all were from all walks of life, different ages, from 20s to their 80s. And we just took a blood sample. And we do something quite cool with the blood samples. We grow them for a couple of weeks into little single cells because we have to study the mutations in an individual cell. We can't just study it in the blood because they're within the cell, and if we just do the whole blood, we won't find anything. So we have to go into a single cell, which you can do where I work because of the technology that we have, and we sequence all 3 billion bases multiple times within the same individual. So these are experiments that render us bankrupt, but they're really important in understanding what's going on in these diseases. Because as I said, right from when we're that fertilized egg, we're acquiring mutations over time and over cell division. And what we can do is then find all the mutations in individual cells, like one of those mosaic paintings, and then we can trace back the origin right to the first mutation of life. And we did that in 10 of our patients. And so what we then draw is a tree. Imagine this is a tree. This is the start of life at the top, and these are mutations. And all we're doing is going, is this mutation unique to that cell? Or is it shared by 2 cells, or 3 cells, or 4 cells? And if you look at, say you have a zoo full of birds, and you know that 5 of them have orange beaks, it's likely that at one stage they had a parent that had an orange beak. You can tell that by shared features, there was a shared ancestor at some point. So all we're doing is using mutations that occur naturally to draw a family tree of blood cells, and we're trying to understand the family relationships between those blood cells. So that's what we do with all those individual blood cells in an individual patient, and we draw a tree. And then we put all the mutations that the individuals have on the different branches of the tree. This is where I lose people, I'm really sorry. But we're drawing a family tree, basically, using mutations, which is a bit unusual. And then the most important thing is, because we know how quickly mutations are being acquired, we can time when those mutations occurred. So we can time the branches of the tree. It's a bit like taking an oak tree and going, that branch there has the Jack2 mutation, and that branch started to grow in 1960. That's what we're kind of doing. And this is the first tree of an individual who was diagnosed with ET at age 21, which is quite young, but it does happen. And each of these dots is a blood cell. And these long branches, it looks like a comb. These long branches tell us that these 2 blood cells here that are normal blood cells, the last time they shared an ancestor, so the last time they had the same parent, now it's not a mother or a father because blood cells have 1 parent that splits into 2. So the last time they had the same parent was at the start of life, right at the top. So all of our blood cells have diverged from one another, a bit like people in different continents. They don't share a common ancestor unless you go back in time at the start of life. But the Jack2 mutation in this individual occurred between 6 weeks post-conception and 1.3 years of childhood. So this was 20 years before their disease presented, and initially we were really shocked, but actually it all starts to make sense, so bear with me. This just tells us that our normal blood cells are making blood independently, and this tells us that the Jack2 mutation occurred early in life. And this, actually, the pattern of branching, can you see it kind of drops and then splits and drops and split? That tells you how quickly the clone, the Jack2 cell, is growing and making daughter cells. And we found the same thing in most people, really. So this individual was diagnosed with PV at age 31. The Jack2 mutation occurred by age 8 of childhood. But other mutations that are common as we get older, they also occur in childhood. So we have this appreciation now in the field of hematology where, as we're getting older, we're all developing these mutations. But it's not that we're developing them when we get older, it's that we're detecting them as we get older. They've been there a long time. But if you have 1 billion cells and it's there and it's in 1 cell or 2 cell or 4 cell, you have to wait for it to grow. And the key thing is, it's a bit like a bank account. If you have a very low interest rate on a bank account, you might not see any growth for decades if you put in £1 or a dollar. But suddenly, if you're at a large number already, the growth becomes appreciable. And it's just a compound effect. So even though these clones are growing really, really, really slowly, it just takes 40, 50, 60 years sometimes to be able to detect it. And once you detect it, it seems really obvious. But it's a bit like those graphs of COVID, right? Those growth curves of COVID. And the same thing happened in this individual. They were diagnosed at age 33 years, and the Jack2 mutation occurred in utero within 4 months of life. And by the time they were diagnosed, they actually had the multiple mutations occurred in utero, and by the time another patient occurred in utero as well, and by the time they were diagnosed, their clone had already acquired 3 separate mutations, each clone overtaking the next one, which is also quite common. But as you can see, there are colored lines in some of the other areas. That's because there are other mutations there, and actually that's what a normal blood profile looks like. And in fact, CALR mutations have been found to occur in utero as well by another group. And little in the UK, I'm sure in the US as well, we do little heel pricks in babies. And if you go looking in patients with individuals with MPNs, the Jack2 mutation can often be found in dried blood spot. In fact, you can do it in the healthy population as well. So it's not just a unique thing. It's just acquiring mutations is a normal part of all of our lives. And it's just a question of what flavor of mutations we're getting. Jack2 can sometimes be second, so there might be another clone already there that had 1 mutation, and then you can get a second event. So that's the only situation where it perhaps doesn't occur as early. And so that's really how long I've had it for. I think I would say that in the vast majority of individuals, the mutations occur early. It doesn't mean that's something to worry about. It just means, and it's actually quite reassuring for me, it means that these things occur very, very, very slowly. So it's not generally because anything happened, because there was a trigger, because, we don't think that, at least, certainly I don't. It's just something that's been there in the background, growing ever so slowly, and it just gets to the point where it's detectable and it's noticeable. So how fast did it grow? We'll last come on to this. So we then took extra blood samples from these 10 individuals to go, okay, we're going to now measure how the clone is growing by just measuring the amount in the blood. And we're also going to look at the pattern of the tree. Are those little splits in the tree happening really quickly? Or are they happening really slowly? Because that tells us whether the clone is growing slowly over 50 years or whether it grew really quickly. And really, so what we have here is all the different clones, and the colors are the different patients, and some patients have multiple different clones, and fitness, this is a scientific term, I'm sorry, percentage of growth per year. This is your interest rate. This is how much it's growing. It's really quite low in the majority of people. It's growing at like 10, 20, 30%. And if it was growing any lower, it would never be detectable for the whole 80, 90 years of life. There are two, so in the majority of patients, it's slow burners. It occurs early, it takes decades to expand, and in those individuals, the MPN generally presents late in life or in midlife. There are 2 routes to clones that are growing fast. They either have multiple mutations because they've acquired the second thing and the third thing, and because of that they've been boosted, there's a bit of acceleration. Or occasionally a single mutation can grow a bit more quickly. And in those situations, you have the individuals who present earlier in life, sort of maybe under the age of 30. And so what we're beginning to realize is the same mutation can grow at different rates in different individuals. And indeed, there's recent data that if we went sequencing our DNA really deeply across the healthy population, at least, we don't even have to sequence very deeply. We just have to look for 1 in 100 cells. 1% of the population have a mutant Jack2 that's at 1 in 100 cells, which is actually quite a big clone. So if you start going down to, is there any Jack2 present? I personally wouldn't be surprised if we found it in everyone. So the question is not, was I unlucky enough to get it? The question was, it was just growing a bit faster in me as opposed to someone else. And we all have our environments that promote the growth of certain things versus not the others. So for us in our research group, the challenge now is how do we work out who's growing quickly, who isn't, and what can we do about it? And this is important because on a basis of a tiny number of patients, you can see 5 dots, this may not even be true. We think that the faster the growth rate, the earlier in life people present. But again, this needs to be validated. So my vision of the future is, if this thing is present so early and it's growing at a constant rate, surely we can do something about it. Surely we can get in early maybe and see who's on a path to a problem in the future and work out how to slow the clone down. But really our job is to try and understand what's happening, and then it's up to a much bigger community to go, right, what can we do about it? Is it worth doing something about it? Will it make a change? Will it make an improvement? So we can look at slowest growing clones, we can try and identify fast growing clones, and maybe we can try and, this reminds me of the sort of growth charts for children. Are they on the right track of growth? As for you weigh the children and see, right, they're on that right curve, which means we're okay. Can we start to maybe do something like that to go, okay, I need to worry about this and this, but not about this. And it changes things for me. Maybe we don't need to get rid of clones anymore. Maybe we just need to slow them down. But these are all like ideas, and we don't know for certain. So in summary, MPNs I hope I've helped explain and not lost everyone along the way, but hope MPNs are caused by genetic changes in our DNA in some key genes that drive blood production. There are variable speeds of growth of some of these abnormal blood cells, but it's generally very slow, which is why these conditions present as we get older. And mutations are generally acquired in the first half of the life, sometimes very early indeed, but that is not something to worry about. That just means that the growth rate is even slower. And its mutation acquisition is really part of our lives and all of our cells in humans and across all species. So understanding what influences these growth rates and slowing them down could enable future preventative strategies. So a lot of this growth work was done by Nick Williams in our lab, and I just wanted to thank our funders on the right-hand side, and I'd be happy to take questions.